inhale oxygen and we exhale carbon dioxide, the waste product of the respiration process. We ingest different food and our body absorbs the nutritive part of the food and the leftovers are removed from the body as waste products. It is not only the food particles, even our sweat is an excretory product. In general, we can say that the removal of unwanted substances from the body is called excretion. We can categorize the excretory waste into two. They are non-nitrogenous waste and nitrogenous waste. Carbon dioxide, water and salts are the examples of different types of non-nitrogenous waste. Ammonia, urea, uric acid, creatine and bile pigments are the nitrogenous waste excreted by humans. Ammonia is the most toxic and readily soluble form. Thus, it requires a large amount of water for its elimination. Thus, the ammonia is eliminated by the diffusion across the body surface. Do you know the organisms excreting ammonia? Bony fishes, aquatic amphibians and aquatic insects are living organisms which excrete ammonia. Here, in these organisms, the ammonia is removed as ammonium ions, that is NH4 plus ions through the gills. The organisms that excrete ammonia is called ammonotelic organisms and the process of excreting ammonia is called ammonotelism. Some other organisms excrete uric acid. So can you guess the different animals which excrete uric acid? Reptiles, birds, land snails and insects excrete nitrogenous waste as uric acid in the form of a pellet, that is a solid waste form or a paste with a minimal water loss. The organism that excrete the uric acid is called a uricotelic organism and the process of excreting uric acid is called uricotelism. The next kind of nitrogenous waste is urea. The organism that excrete urea is called ureotelic organism and the process of excreting urea is ureotelism. Ureotelic organisms includes mammals, terrestrial amphibians and marine fishes. The ammonia produced by the metabolism is converted to the urea in the liver of these animals and releases into the blood and it gets filtered and excreted by the kidneys. The urea is less toxic and requires only less quantity of water for its removal. Out of these three types, which category includes humans? Yes, humans are ureotelic organisms. Now, it is time to check the different excretory structures of invertebrates and vertebrates. In the case of protozoans, poriferans and coelenteroids, they remove the waste materials through their body surface itself. The members of platyhelminths, that is flatworms, and planaria, rotifers and annelids, and the cephalochordates, that is the amphioxus, have protonephridia or flame cells as the excretory structure. Protonephridia are primarily concerned with osmoregulation. In annelids, nephridia are the excretory organs. They are the tubular excretory structures seen in earthworms. Nephridia aids in the removal of nitrogenous waste as well as the maintenance of fluid and ionic balance. In the case of arthropods, especially cockroaches, they have malphizian tubules, which are the excretory structures. These malphizian tubules helps in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands are also the excretory structures and are seen in arthropods, especially in crustaceans. In the members of molluscans, metanephridia are the excretory structures. Echinodermata phylum has a water-vascular system for the exchange of substances and waste materials across the body and surroundings. Vertebrates, including humans, have a pair of complex tubular organs called kidneys. So let us now study the human excretory system in detail in this video. The excretory system consists of a pair of kidneys, one pair of ureters, and a urinary bladder along with the urethra. Recall that the human excretory system is of mesodermal origin, except the urinary bladder. Urinary bladder has an endodermal origin. Let us try to learn each part in specific. First, let us learn the characteristics of kidneys. They are reddish-brown bean-shaped structures. So, where are they situated? It is situated between the level of the last thoracic and the third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Let us examine one kidney in detail. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness with an average weight of 120 to 170 grams. Now, it is time to check the various parts of the kidney. The hilum is a groove seen in the central part of the kidney. It is a region through which the ureter, blood vessels and the nerves enters the kidney. So you can see a broad funnel-shaped space inside the hilum called the renal pelvis. The projections of the renal pelvis are called calyces. Singular we say calyx. Here you can see a major calyx as well as a minor calyx. The renal capsule is a tough fibrous connective tissue layer which provides protection and shapes to the kidney. We can call it as a fibrous capsules also. To learn more, let us classify the kidney into two zones, an outer cortex and an inner medulla. The outer cortex is reddish in color and has a smooth appearance, while the medullary region is of reddish brown color and has a striated appearance. The medulla is divided into a few conical masses, that is the medullary pyramids, projecting into the calyx. The cortex extends between the medullary pyramids as the renal columns and it is called the columns of Bertini. Inside each kidney, nephrons are seen. Nephrons are the functional unit of the kidney. That's all about the general account of the structure of a kidney. Now, we will learn about nephrons and their structure. The nephron has two regions. One is a glomerulus part and the other is a renal tubule. The glomerulus is a network of very thin-walled blood capillaries. Here, the afferent arteriole and the efferent arterioles plays a significant role. The afferent arteriole brings blood into the glomerulus, while the efferent arteriole takes the blood out of the glomerulus. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule and it is called as a peritubular capillaries. A renal tubule has many other parts too. If you check this image, you can see Bowman's capsule followed by proximal convoluted tubule followed by loop of Henle. Then comes a distal convoluted tubule, finally enters into the collecting duct. Now, let us learn about Bowman's capsule in detail. It is a double-walled closed structure enclosing glomerulus. Have you heard of the term Malpighian body or the renal corpuscles? The glomerulus along with Bowman's capsule is called the Malpighian body or the renal corpuscles. And this capsule has a podocyte layer of cells which makes the filtration process more effective. This structure is seen in the cortical region of the kidney. The next part of the nephron is a proximal convoluted tubule or the PCT. It is a highly coiled region seen in the cortical region of the kidney. The PCT is connected with a pin structure named Henle's loop or the loop of Henle. If you check the image, you can see an ascending limb and a descending limb. Recall that the minute vessels of peritubular capillaries also run parallel to this loop and forms a vasa recta. And this part is found in the medullary zone of the kidney. Next, we shall discuss about the distal convoluted tubule, that is a DCT. It is a highly coiled tubular region after the ascending limb. The DCTs of the many nephrons open into the straight collecting tube or the collecting duct. And this structure extends from cortex to the inner parts of the medulla. And this collecting duct opens into the renal pelvis later. So let me ask you a question. Do nephrons are of different types? Yes. What if the length of the Henle's loop is small and not extended to the medullary zone? Such nephrons are known as cortical nephrons. Vasa recta are absent or highly reduced in the cortical nephrons. My next question is, what if the Henle's loop is long and runs deep into the medullary part? Such nephrons are known as juxtamedullary nephrons. Urine formation involves 3 steps: tubular filtration, tubular reabsorption, and secretion. But are you aware of these parts of the nephron where each process takes place? Let us learn the mechanism of urine formation in the next video. Now, let us summarize what we have learned.
Manorama Horizon. Learning made simple.